Okay, well, welcome everyone, and welcome Stuart. Stuart Sandin is a renowned professor at UC San Diego, specifically at Scripps Institution of Oceanography. I've known Stuart for quite a long time now, and he is really a very distinguished biologist, marine biologist, but also a great conservationist, marine conservation, and an administrator. So he wears several hats very successfully. And more specifically, he's a community ecologist, specifically investigating how organisms interact in a complex marine communities. And his research focuses largely on the ecology of coral reefs, with the goal of finding effective management and restoration approaches for this threatened ecosystem. He's also the director for the Center of Marine Biodiversity and Conservation at Scripps Institution of Oceanography. And Stuart, welcome and thank you very much, and we look forward to your talk.
Well, thanks, Nigella, and it's great to see you, and it's great to see everybody who's here. There, I think I have things properly shared now. So thank you for inviting me to come talk to this group and to share some perspectives on what we've been learning here at Scripps about marine ecosystems. I want to start with a little bit of an exercise, if you will, or a shared experience, just to see if we're all on the same page. I want to start by challenging everybody to think about the way that they imagine coral reefs. If we close our eyes, or if we just gaze off into the distance, what do we imagine that coral reef to look like?
Colorful.
They can be beautiful. They can have a whole host of different characteristics. But when I ask people how they see it, I tend to get one of two extreme examples. One of them is people visualizing bright colors, lots of species, a dynamic environment that fills space and is perhaps something that could be remembered in National Geographic covers or wonderful movies and exploratory natural history films. There's a whole nother reality that we oftentimes get, and unfortunately is becoming more common, which is people envisioning death and destruction. They worry about the devastation that some of our coastal ecosystems, in particular in the tropics, have been suffering at the hands of some human activities. We imagine things like coral bleaching or disease or storms tearing away the vibrant organisms that used to live there. This duality of both the vibrance of nature and some of the big challenges that our natural ecosystems are facing is unfortunately two ends of a spectrum, with both being true. What I'm hoping that we can talk through today is some ways that this spectrum is the reality that exists in our coastal ecosystems and natural ecosystems worldwide. But by envisioning just one of those realities, I think that that's one place where we're missing an opportunity. And I hope that we can lean into this to transform our perspective. But instead of imagining what a coral reef looks like at one point in time, instead, I'm hoping that we can start to focus on ways to use our science to shift how we see them today and where they're heading tomorrow. And we'll be using a case study of some of these coastal ecosystems, in particular coral reefs, today. Maybe one of the best case studies was captured by a great friend and colleague of mine, Dr. Jim Porter from the University of Georgia. And he was actually he was ahead of his time in many ways, finding ways that we could visualize coastal underwater ecosystems using photography, finding ways that we could study ecology in a way that was visual and story-based, and using that power of imagery. What he did was he fought really hard on the granting side of things to create a means to follow the coral reefs of Discovery Bay, Jamaica, this on the north coast of Jamaica, during a period of time that he didn't realize was going to be one of the most formative and if not impactful case studies that currently exists in the field of coral reef science. The story began with one of him trying to understand that high diversity of these wonderful coastal ecosystems. We see here, this is a set of imagery. It covers about the size of a big dining room table of a coral reef in Jamaica. And what we see here, all these sticks and brown patches and such, all of this is coral. Coral being the little animals that are building the landscapes in a lot of our coastal tropics. Remember, corals are little animals that have some plants that live inside of them. And one of their special they're like little sea anemones. And one of the special characteristics is that they can build rock underneath themselves. They accrete calcium carbonate, and they create coastlines that create the structure of a lot of these tropical ecosystems. Well, Jim collected this imagery because he wanted to understand all of these different patterns and shapes and structures that you see here are different species of corals. They're colonies. And he looked at these ecosystems trying to think, how are they changing? One year you may have a lot of this Acropora cervicornis, and another year you may have a bunch of growth of this Acropora palmata, another species of coral. And he was trying to understand that dynamic interaction between the different species that lived on a coral reef. But lo and behold, he was in Jamaica during a period of time that got canonized in our literature, describing some dramatic periods of change. It was in the late 70s that we saw one of the biggest dramatic outbreaks of a disease that hit coral reefs of the Caribbean. What you saw in the last panel was a bunch of live coral, and right now all of those corals got decimated. They got hit by a disease that swept through the dominant genera, the types of corals that were there, and ended up leading to a ton of death. Around the same time, we saw a big series of storms hit the north coast of Jamaica, and they broke the skeletons of these corals into the fragments that we see here. Okay, so now he's captured both the way that things looked and how they changed after some dramatic event. You'd hope that we'd be able to watch some pattern of growth. And at some period in time, he did. 1983, here, we see all these little sea urchins around, all the little black spiky things here. These are sea urchins. They're cleaning off this reef. They're making sure that any competitive organisms like algae might be swept away. And hopefully, we're going to be able to watch the growth of these little patches of coral that are here. Well, another dramatic thing hit the island of Jamaica right around this time. And in fact, it was late 1983 that another disease swept through. And this one famously killed over 98% of all of these sea urchins. Just event after event hitting the same island. And now these sea urchins that were cleaning the reef were effectively wiped away. Fast forward 3 more years to 1986. We're looking at the same plot. I want you to recognize this is exactly the same location of coral reef that Dr. Porter went to every year, every 3 years. And what we've seen is this complete overhaul of the entire natural reef ecosystem. Now with those sea urchins gone, there is nothing left to consume all these competitive seaweeds. So the corals trying to grow back now had to compete not just for space and to try to survive through their earliest and most vulnerable life phase. They now had to fight against fast-growing seaweeds that tend to win in these early battles. Let's recall that sea urchins are not the only thing living here, where oftentimes fish can contribute to this whole puzzle. But in the island of Jamaica, we're seeing a population of about 1 million people with some challenges, economic challenges. So the fisheries have been fairly heavily exploited and very few fish remain. So what we're looking at is this almost decimated landscape in terms of consumers. Well, Dr. Porter had brought this work up to us during an academic conference. We've been doing some work trying to study ecological community structure, and I'll tell you some more about that in a moment. And he said, did you know that in the 1970s and 80s, I was doing image-based surveys? And I felt really bad to say I did not know about his work. We were able to find a way to collaborate with him, to go back out as he was retiring from the University of Georgia. He went and he found his first graduate students that he worked with as he was collecting this imagery. And we returned back to the island of Jamaica with him with updated imagery technology. We went and we found exactly the same locations he had surveyed 40 years before. And instead of just using image-based surveys, we found ways that we could combine individual images into these three-dimensional models of what the coral reef looked like. And lo and behold, we found exactly the same location on the planet. We found even the pin that he had marked this with 40 years earlier is in the middle of this coral colony right there. It was the same bit of steel that he had pounded into the reef 40 years before. And we were able to find these exact colonies and then to start thinking, what did 40 years do to Jamaica? Now, we did not find a location that was covered in those big plating corals. But what we did find was a couple of the same corals. What we found when we did look at this, and I'm going to just go back to this, we found in particular this coral colony that had survived for a period of time of 45 years. And when we came back and we talked to our colleagues at the field station there in Discovery Bay, they were flabbergasted. They did not believe that there would be any of their corals left. They had been told that their entire reef ecosystem was gone. They had been told that that everything that we knew about 40 years ago was pulled away from them. Of course, there are a lot of these junior scientists across Jamaica who had seen something different. And for us to be able to spend some time to have some very impactful discussions about what those reefs did look like, what they look like today, it got us started on a conversation about how can we rebuild the reefs of Jamaica for tomorrow. That's just a starting anecdote to get to some of the research that our team has done. I came to the problem of coral reef surveys, and I came to Scripps in particular to address some questions about what's possible in our coastal ecosystems. Now, I'm a community ecologist. What that means to me is that I study groups of organisms that live in the environment. In particular, my taxonomic expertise is fishes. I tend to think about how fish interact in a group of multiple populations and how patterns of ecological competition, predation, and a whole host of other principles lead to the number and types of fish that we may see when we jump in the water in one location or the next. Now, in coastal ecosystems, the fish don't just live by themselves. They live with the organisms that are on the bottom. In the tropics, those may be corals and those seaweeds. In our coastal ecosystems here in San Diego, it may be the kelp and the other seaweeds. So the study of community ecology is simply thinking about how can we understand how many and what types of organisms live in one location, and what are some characteristics of all of those populations that may exist across locations. I came to Scripps with an idea of trying to understand how coral reef communities varied, and we started with this question of how do human impacts, the roles that people play, influence what a coral reef may look like. The year was 2005, if I wanted to get very narrative about this, and we decided to try to visit these remote islands of the remote Line Island Archipelago. Our goal was to describe what a coral reef could look like in the absence of human impacts, so-called baseline coral reefs, what maybe Jim Porter had started to see in the island of Jamaica in the 1970s, but even more so trying to visit some islands like Kingman and Palmyra that had never had permanent human habitation, that had never seen local fishing groups of fishermen working there, and that had never experienced any of the other stresses linked with coastal development. We sought to contrast these ideas of what baseline coral reefs look like with some islands in the Republic of Kiribati, where the Kiribati, the people living in Kiribati, had small, modest human populations in the island of Tabaran of 2,500 people at the time, and the island of Christmas about 5,000 people. And we did this comparative study. Just to be clear and faithful to my colleagues here at Scripps Institution of Oceanography, our research vessel was not one of our research fleet here at Scripps. It was a chartered vessel, and our ships look a lot nicer than what we ended up chartering here in the middle of the Pacific Ocean. It gained me a very early bad reputation for chartering a vessel. It's not quite as beautiful as our fleet, and we've tried to work to make sure that we improve our ship-based resources as time goes on. What we did is we assembled a team of researchers that spanned taxonomic expertise. Me and colleagues working with the fishes, David Obura here and a bunch of others studying the corals and the algae. And we even brought along some microbiologists to look to see what was living in the water. And we wanted to test this hypothesis that human impacts could change what a coral reef looked like. And our hypothesis was more directed than that. We had some ideas of what we expected things to look like. We expected that when people are around, we take out particular types of fish, especially the bigger fish. We know that coastal pollution and other forms of coastal stress could change how many corals live there. And we had reasons to believe that the patterns of pollution and even some multi-step pathways could change the water quality, increasing the microbiological load of the seawater itself. We also sought to understand what truly did a coral reef look like when we hadn't fished these systems. Some of my colleagues, including Jeremy Jackson and Nancy Knowlton, who Nigella know quite well, who were on this expedition, had spent time describing, based on historical records, what baseline coral reefs could look like. But colleagues worldwide didn't believe it because they didn't see very many examples of reefs that had lots of sharks, that had lots of predators, that had the big fish that from before times of rapid human population growth. Now, not quite as many photos and not quite as compelling, but probably pretty well repeated, ended up being an output from this expedition. And the photos I'm referring to are this 8-panel slide that is probably the most cited work that I have in my life. For all the work that I do, understanding the models of ecological community structure, population biology, and the reams of data our team has collected, I think most people on the planet know me for this 8-panel set of photographs, none of which I actually I collected myself. So I sometimes have some challenges in making sure that people know that I am an ecologist and not just a photographer by discipline. But this 8-panel slide was a way for us to tell the story of what we learned across the 4 of these islands: Kingman, Palmyra, Tabaran, and Christmas, what these 4 islands look like. Challenging this idea of what can a reef look like and how does it change with human activities. If we look at the left-hand column here, this describes what the fish assemblage looks like. When there's no fishing, we get more fish. Nothing there should be surprising. But what was surprising was the density and sizes of some of the predatory fish. We actually were able to document locations where there were more predatory fishes than there were prey fish. so-called inverted biomass pyramids. The right side of these photos. These 4 photos are describing what the bottom of the reef looked like, much like Jim Porter described in the 1976 imagery. Lots of corals growing, and the whole landscape really just dominated by the organisms that are creating the coral reef, through to systems that are dominated by seaweeds. Seaweeds don't create islands and atolls. Instead, they tend to dissolve those islands and atolls. And none of this and all of these things met our expectations. If we start disrupting these ecosystems, we go from baseline, productive coral reefs that are beautiful, that are wonderful, that have high diversity, high mass of fishes, and high growth of corals, through to systems that have few fishes and that have more seaweeds than they have the calcifying organisms. Now, we put a lot of emphasis on the fish, and also getting close to our Halloween holiday season. I want to highlight this fish, which we sometimes call the vampire snapper. My great colleague, Dr. Jennifer Smith, took this photo from that Line Islands cruise because this big predatory snapper was just so curious to see her during this expedition, she had to push the fish away with her camera lens. Along the way, she pushed the button and captured a photo. I highlight this simply to say that the study and the output from this work, the conclusions that we came to, really were built upon a pretty straightforward narrative. That narrative being locations that have no people can have lots of big fish. Locations that have no people can have lots of corals. And locations that have people see the loss of each of these elements of a productive coastal coral reef community. Okay, we finished that expedition. We did some additional work in the Line Islands, and at one point, working with some colleagues from an NGO called One Reef, and with some leadership from the Federated States of Micronesia, I got an opportunity to visit some of the islands in the state of Pohnpei, in that country, the Federated States of Micronesia. We did some surveys working with the traditional leader of an offshore island. This is Pohnpei Island in Pohnpei State of FSM. This is Ant Atoll over here, and then we have Paquin Island up here. The state of Pohnpei in Micronesia actually has dozens of islands, but these are the ones closest to the main population center. We visited this island of Ant Atoll at the invitation of the traditional leader, the Rosa of that island, to say, what do the reefs look like? And we came up with imagery, data, but more importantly, stories that really blew my mind. We saw coral reefs that look like this. And if this looks like one or the other of that spectrum of coral reefs that Dr. Porter shared with us, the one with lots of corals or the one with lots of algae, the before time or the time after a bunch of catastrophes hit Discovery Bay of Jamaica, I would put this reef squarely in that before times, in that intact, impactful, and beautiful coral reef settings. But the interesting part of this story was that this coral reef, we were there to investigate, to study, and to spend some time working with the Rosa, because he claimed that the island of Ant Atoll, had been overfished, that it had been overrun. And him, when he had assumed the role of Rosa, which again is analogous to being king of a big section of the Pohnpei community, he was trying to reinforce some rules of traditional leadership, trying to get the people who wanted to fish on Ant to do it after getting permission from the Rosa. He wanted to limit the fishing such that whenever there was a festival, some big celebration, that he could allow the people of the community to fish in a giant supermarket of reef fish, that being the fish of Ant Atoll. And he wanted there to be an opportunity for there to be a vibrant coral reef community for the generations ahead. Now, he was looking at this reef setting as one that needed help in protection, and that's what our colleagues at One Reef had started to work with. And we at Scripps were providing some of the biological, ecological support, some data advice. And it really, it threw my narrative of people only shifting coral reefs in a negative direction, in a completely different direction. I realized that we could find a way to manage a coral reef ecosystem and to support the needs of humans. And it all came down to one simple principle. It was in finding leadership and community, much like the folks here in Micronesia. This is the Rosa, and this is Rose Bain, his wife and second leader of Ant Atoll. Their leadership and the leadership of the folks before had maintained a coral reef for the community by the folks from the Conservation Society of Pohnpei, that's who these folks are, had found some ways to maintain a balance between the needs of the local community and the viability and livelihood of the coral reef itself. This threw my head spinning a bit. And it made me realize that our simple narrative we had built in the Line Islands was just that simple. And it didn't account for the reality of human engagement. And it didn't allow for that reality of the dynamic nature of a coral reef ecosystem itself. It was based in large part on the relationship I built with Rosa and the community in Micronesia that we launched a new research thrust. So in 2014, my team began an effort that we called the 100 Island Challenge. This was us taking a step back in eating a slice of our humble pie, as it were, to try to start thinking about what does lead to the variability of coral reefs worldwide. We realize it's not as simple as if people are there, it doesn't work. And if people are there, it does, or sorry, if people are not there, it works. And if people are there, it doesn't work. It's not as simple as saying that we have the same expectation for every reef. We know there are multiple things driving what's there. So we sought to, rather than put our a priori perspective of what should and should not exist in the world into a Scripps-like perspective of learning from the variability that exists across our world's ocean ecosystems. And with that, we sought to do just what the name says, to visit, to study, and to work with colleagues from 100 islands worldwide to really conduct this latitudinal survey to understand the variability of our coastal ecological communities. The geographies have been expansive, and the project as it stands involved work in about 19 different countries. The total number of islands we visited is about 123 right now. We've had some challenges. There was this little virus that went around the world for a bit. You might have heard of it, that provided some challenges in our efforts to visit each of these locations, especially since COVID was striking a lot of our Pacific Island community much more strongly than it did a lot of the coastal, the continental communities. But we've had some very great successes across these geographies, mainly linked in the Pacific, but going all the way to the Western Pacific of Okinawa and Japan and Taiwan, the Indian Ocean across the Maldives, and with a series of geographies across the Caribbean. Our target was to visit each island at least 2 times, so we were able to study not just what the reef looked like, but how it was changing through time. Our study was relatively straightforward. It was following protocols that we had developed through a decade of decades of field experience, which was to try to find an efficient way to take our Scripps scientists, our partner scientists, drop them in the water, and then provide a synoptic snapshot of what that coral reef ecosystem looks like. We can see here in this video a snapshot from one of these 360 cameras as it were, showing what we may do during one dive. Put 4 or so divers in the water. Find ways to get yourself settled and safe, and then send some divers in different directions. We have people who are trying to document what the fish assemblage looks like. How many fish, what species, what size. We have people looking to see what's on the bottom. How many corals are there, how much algae. And we found ways to capture imagery that provides us a means to follow not just what's on the bottom of the reef today, but for us to be able to take images and then do that same form of imagery a couple years later so that we could watch what's changing and growing. The work did involve these studies of fish, and if we focus on that, we've been able to learn some basic principles. We shouldn't be surprised in this to see results that look something like this. For a subset of the islands that we studied, we've been able to show some patterns like this. And let's just orient. We're looking at how much fish there is in a location. We do that by estimating how much they would weigh if you were to take every fish, pull it out of the water, and then weigh them. We do this without killing them and weighing them. We do this by swimming down a line and counting how many there are, what species it is, and how big it is. We then separate these islands. We can test that basic hypothesis we had at the beginning of the Line Islands work. Islands that have no people living on them are unshaded. Islands with people living on them are shaded, or do have the shading. And we're able to document some basic principles. Okay, by and large, if nobody is fishing in a location, you tend to get more fish. That should not be surprising. More so, what we see is that the particular fish that are around, as documented here in the Pacific remote island areas, this dark band here are the fish like that snapper, that vampire snapper I showed you earlier, and some of the sharks. That's all of this biomass. By and large, you get more predatory fish when you don't fish. And that's just the foundation of fishing. If you put a fish, a little bait fish on a hook, you drop it over the side of a boat, it's not going to be something that eats seaweed, something that eats plankton, or something that eats a crab that's going to come and bite that hook. It's going to be a predator. So our fishing practices will catch predators first. But I want to look at another element of these data, which is to say, how consistent is the signature? We see that there's some variation, even in the inhabited islands, of how much of this biomass or how much fish there is. And that variability is linked to the management. Much like the Rosa from Ant Atoll in Micronesia, the amount of management that we have here in Kiribati, or in some of the islands of Hawaii, or in the Commonwealth of the Northern Marianas, influences what that fish assemblage looks like. In the balance of the geographies where we've worked, we've seen a whole host of variability of how much fish is there. That's due to the activities of people and how we are conscious in how we use, preserve, and consider our fish for today and for tomorrow. We look as well at the bottom, and I want to give a sidebar here on ways that we look at what's living on the bottom. Over the last few years, we've invested quite a bit of time, and this has been one of the beautiful elements of being a professor at UC San Diego, is this ability to work across departments, not just here at Scripps, but with colleagues at the Jacobs School of Engineering and such, to try to think about what technologies can be there. We've found ways now to not just study the bottom of a coral reef using humans conducting surveys, but instead to use imagery, photography, and in this case, composite photography, to create these landscape level views of what a coral reef looks like. We have views that capture large areas, about the size of a tennis court, of coral reef that we can document what lives there. But then using this ability of working with quote unquote digital twins, we can zoom in to see not just the overarching view, how much coral is there, but we can study some fundamental things like how many coral juveniles are there, What's the size structure of those corals? How many are there? And as we think about this, how it may change through time. This technology is founded on some basic technology that's around. Many cell phones and cameras today will create composite views. Think of the quote unquote landscape imagery that we can do with a cell phone today. This technology is analogous to this, except we're able to do this in a full, to create a full 3D reconstruction of a coral reef. At one point in time, we can make some very basic studies of what lives on a coral reef. Take this landscape view, again, the size of a tennis court. We can map out what corals are there, different colors. We can think about how many there are by counting how many of each color there are. And we can think about how they're aggregated or disaggregated. These are fundamental ecological studies that have led to us really understanding a lot more about the basics of colonial organisms. Corals are colonial organisms in that they just live with one another. Some really fun ecology that we've been able to do. We can look at the same landscape and look at where all the coral babies are, the juveniles. This is the next generation of corals that we can zoom in and zoom out from this imagery to collect some really profound ecological insights. And we can even do some things that I never thought was going to be possible. We can look for patterns of seaweed. Okay, this sounds strange, but this has been part of the exploratory science we've worked on. We mapped out how much of a particular species of seaweed there was across a landscape. And if I quote unquote turn off the rest of, or this is the seaweed, Halimeda, and if I turn off the background, I end up with a pattern that looks like this. The two colors here are 2 different years. But we see this emergent patterning. We see something that gives this polygon-like pattern that just emerged out of a landscape. The seaweed's about the size of my finger, but these patterns, these polygons, are at the scale of 3, 4, 5 meters. This is emergent patterning. This is one of these really fascinating elements of complex systems where you can get a pattern that happens at much larger than the scale of the organism itself. Fascinating complex system science and mathematical biology here, but it's something that we could only explore through these partnerships of employing technology for other domain sciences that don't typically have access to this type of imagery. Our director here at Scripps, Dr. Margaret Leinen, deems this conservation technology, and I think that term makes a lot of sense. Using technology for goals that go beyond just developing profit, but instead getting into the hands of practitioners for the management of our natural resources, for the betterment of our natural resource stewardship for tomorrow. But even more important, and maybe most exciting for me about this technology, has been our ability to see through time. Hopefully this video is not too jerky for you, because I sometimes worry about Zoom. And Nigella, does it look okay and it's not making you seasick or anything? Terrific. What we're seeing here is a 4-dimensional view of a coral reef. It's a 3-dimensional model in every point in time, but we're looking at the same point in the coral reef at different points in time. You can follow this little point right here, another measuring stake or piece of steel we put in the reef, and we can just follow it from one year to the next. And we can start to do something that we never had the ability to do before, to watch what's changing underwater rather than just think we see what's changing. We can now look at one point in time and then see through time how corals grow. Yeah, we can watch them die as well. We can watch them split, but we can watch all of the behaviors that these organisms have on the bottom of the ocean. A fish may spend its entire life living under this hole, and that fish may, if it could tell us stories, could tell us what's happening with the corals around it. But unfortunately, those fish aren't talking to us, and we haven't had this ability to see underwater and to use the tools like we've been doing with Google Earth and other landscape-like mapping tools on land. We've yet to be able to do that underwater. Watch this, for instance. This coral got hit by a big storm, got flipped over. So this is it now in 2015. but now 2016 covered in calcifying seaweed, and moving on. We watch different elements of these branching corals growing and filling this landscape. We watch this thing bleach partially, but then regrow. We're seeing that dynamic nature of the bottom of the ocean that we only tried to infer before, but now we can watch. And if we can watch it, we can start to use that knowledge of what's possible to manage for tomorrow. We can watch systems like this area of Moorea, where everything that's green here is new coral that wasn't there the year before. This landscape grew so much, you could, if you were to weigh how much this new coral in this tennis court sized area weighed, it would be as much as like a full truck that was filled with rocks. This is a fascinating amount of new coral that grew here just based upon fundamental ecology, just population biology. But we've never been able to watch this before. And we can zoom in on particular areas. We were recently in the Line Islands, and we went back to a location that we wanted to study. And we want to collect another one of these images from a massive coral, this big coral here. And we showed up and we said, oh no, that marking stake that we have is gone. So we put in a new stake. That's this piece of steel you see here. We said, okay, well, this is fine. It must have fallen out and we have to replace it. Well, I went back to the old imagery that we collected 4 years before, and I found where the stake was in this same coral colony. And then I looked back, and that stake was actually still in the coral. If you can see this little pink dot right there, that's this stake that was sticking out of that coral 4 years before. I show this not to try to reveal how bad I am at finding these marking stakes, but instead to think about how fast this coral colony grew. We never knew that corals like this could grow upwards of 15 centimeters upwards in a simple 4 years. That is a massive amount of new rock that's being generated. And it's only because of new technologies we were able to find these spots. Or even these landscape views in the southern Line Islands of a coral colony that we had seen. This is dead right now, but then we went back to this 4 years later, and this is what that same colony looked like. It was covered in all these new corals, and again, only because we had this ability to find the same spot underwater and then image it, where we're able to get this view of what's possible from a coral reef. It can die, but it can grow back. And when it grows back, it can grow back different and better and stronger. And this is something that I believe that our colleagues in Micronesia recognized, that if they lost some corals, they could grow them back. But now our scientific community was catching up. We're finding a way to document how things can change and how we can see our coastal ecosystems as dynamic in nature, as having this ability, this resilience to regrow after some disturbance event. So the 100 Island Challenge has been a wonderful campaign, and it's one that we're still spending our time unpacking the data. We visited, as I said, over 120 islands, and we've been able to see, if nothing else, that coral reefs are variable. We can go for a tour now. We can use this ability of immersive technology or digital twins to take you from the Solomon Islands through to the islands of Hawaii, and now down to Tonga. We can look at how one reef looks different than the next. And right now, my team of students, staff, and increasingly undergraduates are able to explore these geographies from across the globe to be able to think about what is happening ecologically across the world's tropics. But even more so than just our team here at UC San Diego, we're using this immersive technology to work with our colleagues from across these 19 countries. We're working with the colleagues in Micronesia, in Okinawa, in Taiwan, and across the Caribbean to employ these similar techniques of image-based coral reef surveys for us to update our management worldwide. Working with our colleagues at One Reef, these were the folks that first introduced me to the ROSA, and where we worked together in Ant Atoll. But they've actually expanded their effort. They're a non-governmental organization founded on the principles of working with community groups to provide technology as needed, access to insights into some of the legal frameworks for coastal protection, and some access into educational platforms for revisiting some of these community-based management targets. We've been serving for the last few years as the academic colleagues for this, both through my lab at Scripps and through our Center for Marine Biodiversity and Conservation, to provide access to not just the data, but the technology, where our colleagues now in Micronesia, in Polynesia, and now in Fiji are collecting the coral reef survey-based data and applying these insights from community ecology. What we have is a worldwide group of stewards. The folks are super knowledgeable about what's happening with their coral reefs, but haven't had access to some of the scientific tools that exist. And so we've been spending a ton of time to leverage the human bandwidth that exists worldwide for the management to happen, not based upon scientists from Scripps showing up saying what works and what doesn't, but instead our colleagues from UC San Diego providing access to some of the more expensive technologies, but making it not expensive, doing the R&D and handing it off, and making sure that our colleagues across the globe are all working with the same storytelling capabilities that technology has. And I'll tell you one thing, I've been learning from our colleagues from across the Pacific into how to tell some of these stories about how these reefs work, because they're the ones that see through this, and they're the ones that are helping us to understand not just what a reef looks like today, but to visualize what it can look like tomorrow. Nigella, if I have about 5 more minutes, I'd love to tell one more anecdote about the way that we can couple this community ecology with some of our global partners.
Yes, please do. I'm finding this fascinating, so please do.
Great. So through our Center for Marine Biodiversity and Conservation, we recently had a great opportunity to work with some colleagues from an organization called Island Conservation. Island Conservation has a legacy, a decades-long legacy of working with island ecosystems and trying to help them to improve and restore the island ecosystems on land. A big part of their portfolio is to manage and to remove invasive species, rats, rabbits, goats, different animals that were brought in intentionally or unintentionally that could have devastating impacts on islands when left there. This example here from Chile, an offshore island that had rabbits, those rabbits had no predators, ate all the vegetation and left the system denuded. Well, very soon after a campaign to eradicate those invasive rabbits, you ended up with a landscape that looked like this. Some additional restoration efforts of planting some of the, providing some seed stock and such, and we ended up with an ecological community that was thriving again. What, you may ask, does this have to do with the ocean? Well, we can generalize this, and we can show that there's a whole host of things that can happen on land when you have an invasive pig or rats or goats. And what happens when those invasive animals are removed? You get more vegetation. You get more of these charismatic vertebrates, let's say. Seabirds tend to show up in droves, and the vegetation gets more complete. Okay, we know very well from case studies worldwide, this is all the invasive mammal eradications from across the globe. And reviews of those successes have shown just vibrant rejuvenation of the terrestrial ecosystems. But if we go back to what my colleagues in the Pacific have been teaching me, they never thought about the land by itself. Like this model of the Ahupua'a from Hawaii, it was all management of islands from Pacific Islanders has always been a ridge to reef model. This idea of what's happening, the use of resources up in the mountains, in the flatlands, in the coastal ecosystem, and then into the ocean is all linked. And they're typically linked in the Ahupua'a system in Hawaii, at least, through watersheds. You've got one river, and that's an entire management unit. There was this view that what we're doing on land is going to influence the ocean. By returning to that knowledge, by working with some of that historical human knowledge and thinking about some of our modern management efforts, we started thinking, well, shouldn't we be thinking about, in this island conservation sense, what's happening to the adjacent ocean? Shouldn't we be building some hypotheses about how an eradication and a restoration effort on land could and does influence our ocean? And shouldn't we be using that as one of our targets with our colleagues worldwide in emphasizing some of these opportunities to improve our coastal landscapes? We know that in some cases, if you have a system, as say in this case, invasive cats can actually lead to some of our seals getting diseases. It can lead to there being fewer birds, but in a system that has the birds, that has the seals, you get a more vibrant coastal ecosystem. The nutrients from seabirds come through and fertilize the reef. Or in a system that's more temperate, we get more birds, leading to the different interactions with our intertidal organisms, shifting the entire landscape of the seaweed, the bivalves, and all of those fundamentally important marine organisms. Their populations are intimately linked with the structure on land. We recently started a campaign in partnership with these colleagues from Island Conservation, an organization called Rewild, and then our Center for Marine Biodiversity and Conservation here at Scripps, called the Island Oceans Connection Challenge. It's perhaps one of the broadest campaigns we've had in the island-based or the community ecological effort, where we're putting the an academic organization in partnership with NGOs to be providing explicit academic insights into what does happen across this gradient from invasive species to eradication, and how does that influence the coastal ecosystem. We began by reviewing literature that's out there and trying to build some view based on the knowledge from these complex interactions of land to sea connection, groundwater exchange. We've hosted workshops here at Scripps to bring together partners from traditional leadership through to NGOs and academics. But more importantly, we've begun a campaign where, in partnership with the folks from Island Conservation and Rewild, who are working with national governments and traditional leadership to realize campaigns of coastal management, we've been using this as a set of ecological experiments at the grandest of scales that we're documenting how these islands are changing from land to sea. We're trying to put some current numbers and some documented data onto this puzzle that complements the way that humans for generations have thought about how we watch landscapes change. We're working with the traditional knowledge to put in some of our current ecological knowledge and to try to marry the two for us to build a better set of expectations for not just what do things look like today, to not just lament how things have changed from yesterday, but instead to start to design in a meaningful way what we want tomorrow to look like. And we know that we have some levers of management. We know that we can influence how many seabirds are around through some control. But now we need to start thinking about how does this all play together with some of our elements of marine stewardship, of resource stewardship, and of a lot of our community-based needs. What we've started to do is to really take that leap, to move our ecological sciences from one of trying to be building foundational principles where we've tried to, as a discipline, mimic some of the sciences, the fundamental, build a periodic table of ecological principles. But every time we do that, we realize that every island, every coast and every mountain that we study has its own characteristic. That's why we call it natural history. It's these stories of what our globe is doing. For us to manage this, we have to think about not just those fundamental rules, but we have to be using the experiments of change that are happening today as an opportunity to learn, but then in real time to apply that knowledge so that we can manage for a better tomorrow. The partnerships that we've been able to build through our Center for Marine Biodiversity and Conservation, the opportunities to work globally, like are enabled here at Scripps Institution of Oceanography and UC San Diego, and just some of that trust that we've been able to maintain with our colleagues globally is just providing us this most unique opportunity to face tomorrow with our eyes open, with this ability to learn and to share, to educate globally, and to hopefully build something where what our tomorrow looks like isn't something that we believe is a fait accompli, but instead something that we have agency in, something that we can put our finger on, and that we can help to influence the trajectory. And as I look out here with our growing undergraduate population here at Scripps, and when I work with our UC San Diego students across the board, all they're looking for is some reason to start thinking, how can I use my science to make tomorrow slightly better? In the natural resource sense, it is by building communities, by being smart scientists, but then also being able to listen to different knowledge bases and to start going out on a limb and providing advice on where science helps to teach us what tomorrow can look like. And then hopefully some of our colleagues will also get into the fields of government, governance, and advocacy. We aren't on the advocacy front, but our students will go all across the board. We all vote, and we have all these opportunities to face tomorrow differently. So I've been really pleased to work in our community here. I've been really pleased to work with our partners worldwide. And I really appreciate the time to share a little bit of this science with you guys today.
Stuart, thank you so much for that talk. I can't tell you how much I love your passion and this enormous body of knowledge that you have made with so many partners. So it's not just your students and other folks at Scripps. It's not only the whole of UCSD, but so many other people around the world to create such a knowledge base. I think we've pretty much used up a lot of time for questions, but we might have a little time for a couple of questions. Anyone like to ask Stuart a question? Well, in that case, I have a few questions for you. I think, one of the things you obviously didn't talk about is one of the main disruptors of coral reefs today, and that is coral bleaching. Mm-hmm. And, you talked a lot about what you've learned about the dynamics of reefs and how very changeable they are over short periods of time, and also that they can go back quickly. Does this give us some hope for renewal after a lot of the bleaching we've seen recently?
Yeah, that is the million, billion, whatever number of dollar question. Right now, it's certain we can view the problems for coral reefs in a way that's similar to most of our natural ecosystems, as built upon two different types of stress. You got the local stresses, like we pointed out, the fishing, the pollution, the trampling, and you have the climate issue. I would argue that each of them has an equal amount of profundity in terms of changing the ecosystem and really forcing it. And both of them have to be addressed for us to be able to start managing into the future. When it comes to corals, they are one of the frontline organisms. We have documented very, very in much detail how temperature can disrupt some of these symbioses that they have. That's the coral bleaching and the coral death based upon hot water events. We're finding some things where intact systems, places that don't have local disturbances, can face the hot water events where the corals will be able to survive or to regrow rapidly if